Apologize for the lack of cute animal videos or pictures. There are some pictures, but it's gonna be a bit more dry than what you just had. So I don't know if this was a good order, especially with the lack of coffee. What I can give you though is, I would say, interdisciplinarity. So for the past few years, I've been working in developmental psychology departments, but I have a background in cognitive science and computer science. And now I'm at IBET, which is the Institute for Biodiversity and Ecosystem Dynamics. So I've been hopping around quite a bit. And in my presentation, there will be a lot of human and non-human examples. So this might explain where this is coming from. So what I'm going to do today is present you some of my dissertation work. Specifically, I'm focusing on the evolution and development of sensitive periods in changing environments. And I will start by just defining a lot of things and setting the stage. Okay, so all individuals are shaped by their experiences. And the time periods during which the impact of experience is greatest are called sensitive periods. Sensitive periods exist across the entire tree of life and play a crucial role during development. These sensitive periods are periods of heightened phenotypic plasticity. And plasticity is the ability of our genes to produce changes in our bodies and behavior, our phenotypes, based on experience. Ontogeny refers to the time period during which experiences can have an impact on the development of a specific trait. Although sensitive periods appear to be more common early in ontogeny, they also exist at later developmental stages, such as adolescence. Across species and traits, interest in such later sensitive periods has grown vastly. Empirical studies suggest, for example, that some personality traits in rodents are shaped during adolescence, that human adolescents are particularly sensitive to social feedback, or that mites, or more specifically bulb mites, show plasticity in response to nutritional conditions towards later developmental stages. These examples illustrate the variation that exists in the onset and duration of sensitive periods. And this variation exists at different levels of biological organization. It exists, for instance, between traits within a single individual. It also exists in the same trait between individuals within species, so individual differences, which is something I'll be coming back to later. And it exists between species. But what explains this variation in the timing and duration of sensitive periods? Over the past decades, we have made tremendous steps towards understanding the neurophysiological mechanisms underlying sensitive periods. And understanding this variation, at least I think so, is key to manipulating when and how organisms are shaped by experiences. An extreme case of such manipulation would be to reopen sensitive periods for specific experiences. So to speak, to turn back the developmental or the learning clock. And to date, neuroscientists have developed experimental interventions which can erase signatures of early adversity in rodents. So just the prospect of achieving something like this in humans is just very, big. Aside from neurophysiological processes, the timing and duration of sensitive periods also depends on properties of the environment. For example, do we expect children who are exposed to caregiver unpredictability to lose plasticity at a similar age as children who experience consistent parenting? At present, we know actually relatively little about how environmental factors shape sensitive periods. In the past decade, there has been a search and mathematical models that explore under which environmental conditions we would expect sensitive periods to evolve. And like maps, these models help us zoom in on some factors and processes while leaving out others in order to navigate this complex space. They often present a much simpler version of reality. Not despite, but because of their simplicity, we've actually learned quite a bit about the evolution and development of sensitive periods. So we have learned, for example, that plasticity depends on information about the environment that is available to developing organisms. Specifically, models have found that plasticity tends to be higher early in ontogeny, the higher organisms' uncertainty about the environment at the onset of ontogeny, and the better experiences during development help to reduce this uncertainty. Models have also found that plasticity tends to decline more slowly when experiences are noisy and do not provide as much information in order to reduce uncertainty about the environment. These models have predominantly found sensitive periods early in life. And one explanation for this pattern may be that the majority of these models have explored environments that are stable within a lifetime of organisms. Under these conditions, there's typically no need for organisms to maintain costly plasticity. But as we know, most long-lived species, such as humans or other mammals, are likely to experience changes in environmental conditions throughout their development. And these changing conditions may favor sensitive periods later in development. Today, I present two models from my dissertation which formalize these ideas. Model 1 explores how changes in the quality of information within an organism's lifetime shape sensitive periods. So why might we expect such changes in the value of information? It could be, for example, that certain experiences or cues which are able to transmit information are more abundant or only available at some developmental stages, but not others. For example, mothers transmit information to their developing fetus about the expected long-term nutritional environment after birth. And this information is unique to that life stage. It's only available in the womb. Information that the newborn receives outside the womb tends to be less informative about this long-term nutritional environment. So in this example, the quality of information is highest at the fetal stage and then subsequently declines. In Model 2, I look at how changes in the environmental state itself shape sensitive periods. Changes in climate or seasonality might cause environmental fluctuations, and alternatively, the organism itself may move through different environments and habitats across its lifetime. For example, in humans, moving within our countries may drastically change the environments that we experience. Okay, I will now proceed by providing even more technical details about the models, so please bear with me. In Model 1, I explored variation in the quality of information, while the environmental state itself remains stable. So organisms can sample information by sampling cues throughout ontogeny, and the quality of that information depends on what we call the cue reliability. The reliability of a cue essentially indicates how well the cue helps the organism to discriminate between different states of the world. The cue reliability can range from low to high. To make this a little bit more concrete, just try to think of a smoke detector. If you have a highly reliable, very expensive smoke detector, it will only go off in case of a fire, but not due to a steamy shower. So in this case, this smoke detector can very well discriminate these two different states, and when you hear the sound, you know it must be very likely a fire. As indicated earlier, the reliability of cues may change across the lifespan for different reasons. The one reason that I mentioned before was that cues may be more available in some developmental stages, but not others. Or it could also be that the organism's ability to detect cues changes across the lifespan, for instance, as their sensory systems mature or degrade with age. In Model 2, I explored changes in the environmental state itself, while the cue reliability remains stable. Here the environment fluctuates between two discrete states, and the rate of this change is a parameter in the model. Therefore, I can explore a range of environments from relatively stable to rapidly fluctuating. Both models extend the previous model of sensitive period evolution, and one important assumption of this model is that development is both gradual and irreversible, and I'll come back to that point in a second. Okay, I'm going to try to walk you through the basic setup of the model, hoping that this will make the results easier to understand. So at the start of ontogeny, organisms are born into one of two possible states of the environment. Just for purposes of illustration, let's assume those are safe or dangerous. In the beginning, organisms are only equipped with a prior estimate over the distribution of these states. And the prior can say, for example, that both states, safe and dangerous, are equally likely, or that one state is much more likely. So maybe it's much more likely to be in a dangerous environment. In each time period, the organism then samples a cue about the environment, and that cue can say, It's safe or it's dangerous. And remember that for a specific environment, there is this Q reliability that specifies how much information the organism can gain from this Q. With all of this in mind, organisms can update their estimate about the environment via Bayesian inference. And then they get the opportunity to adjust their phenotypes. And to adjust their phenotypes, they're given three options. Either they can do nothing, so they just forego specialization and leave everything as is and continue. Or they can specialize incrementally towards a safe environment. For example, they could grow slightly bigger ornaments to attract mates, or they could incrementally specialize towards a dangerous environment, for example, by growing bigger weapons to fight. And I'm stressing incrementally because developing these things in the model takes time. So it's not like within just one time period, the organism can fully develop any of these phenotypic specializations. So in that sense, development is incremental. Once such an adjustment to the phenotype has been made, it can never be undone. In that sense, development is irreversible. However, an organism that had previously specialized towards one type of environment, so it started to invest in ornaments to attract mates, can later on still invest into weapons. The cycle continues until the end of ontogeny. And then next, organisms accrue fitness based on how well this phenotype that they spend time building matches their environment. And the underlying idea here is that the better the phenotype matches my environment, the more likely I am to survive and the better my chances at reproductive success. So with this rather simple setup, I can compute optimal developmental trajectories for different combinations of priors and Q reliabilities. And these so-called optimal policies can be understood as a blueprint for development. So for each possible state that the organism could be in, it puts out the optimal phenotypic decision in that state. To compute these optimal policies, I use a method called stochastic dynamic programming. And no worries, I'm not going to go into details about that. I just thought it might be nice to tell you that it's a method that takes a probabilistic and a problem with probabilistic outcomes, and it breaks it into smaller subproblems, finds solutions for these subproblems, put them all back together in order to solve the bigger problem. If you're interested in this method, I highly recommend the book, a very old book in the corner of the slide, but it's actually very nice, nice tutorial for the method. And 2 years ago, I also gave a workshop on this method, and there's a link to the materials for that if you're interested. So finally, from the optimal policy, I can simulate entire populations of organisms and their developmental trajectories, and then with just one more step in between, I'm able to quantify changes in plasticity across ontogeny in these data. So after all this effort, I get curves like this. I'm not going to go into detail about how I'm going to quantify plasticity, but if you're interested, you can ask me later. I'm happy to elaborate. Okay, just as a reminder, so we're all back on the same page. In the first model, the reliability of Qs can change between consecutive time periods. So in one time period, it might be a certain value, and then in the next time period, it could be a bit higher or lower. In the other model, the Q reliability remains constant. However, between two time periods, the state of the environment may change. So it doesn't have to, but it could. So it could be that the organism is first in a safe environment, then again safe, then dangerous, safe, and so on and so forth. Okay, with this basic setup in mind, I'm finally going to present some of the results, and I'm going to start with the first model. So I've explored 3 different patterns of change. So the reliability of Qs may continuously increase across ontogeny, so start out barely reliable, and then at the highest point, Qs are really reliable. So if you see a Q and it says the environment is safe, then it's very likely that it's actually safe. Then I explored another pattern where the Q reliability first increases and then decreases, resulting in this triangle, hence the triangular shape. And lastly, I explored a pattern where the Q reliability continuously decreases across ontogeny. I found sensitive periods towards the middle of ontogeny, potentially corresponding to adolescence in some species, when the reliability of Qs increases at least across some portion of ontogeny. So that is true for the increasing in the triangular pattern. When the reliability of Qs decreases, I only find sensitive periods at the onset of ontogeny, which makes sense because under these circumstances, organisms have access to this highly reliable information early on, so there's no need for them to remain plastic. They can just go all in into specializing towards that environment. I mentioned earlier that some empirical studies suggest that adolescents may be particularly sensitive to social feedback. According to my model, sensitive periods at later developmental stages may occur when the reliability of experiences increases at some portion across development. So perhaps a concrete example for this is social feedback as a cue to mate value. Social feedback from the opposite sex may be more informative about your own mate value during reproductive years than in childhood or at old age. So this is very hypothetical, by the way, but this just illustrates how very general models like mine may be used to generate specific testable hypotheses. So let's have a look at the next model, where I explore changes in the environmental state. Here I find sensitive periods tend to evolve towards the end of ontogeny when the environment fluctuates frequently. This finding contrast results from models assuming stable environments. It indicates that natural selection may heighten sensitivity to cues towards the end of development when the environment changes rapidly and organisms develop incrementally. This makes sense. When the environment changes, cues towards the end of ontogeny should be better predictors of whatever comes after that life stage than cues early in ontogeny. And this is also something we observe in empirical patterns of behavioral and phenotypic plasticity in some species. Going back to my bulb mites from earlier, in bulb mites, nutritional conditions during the final developmental stages strongly determine whether males mature as fighters, which you can see on the left, so they are bigger and they have this extra fighting leg, which I circled in red, or benign and defenseless scramblers, which you can see on the right. When the environment changes slowly, we also observe that sensitive periods evolve midway through ontogeny. In this model, this happens when experiences early in ontogeny contradict organisms' estimates of the environmental state. This pattern has also been found in other models of sensitive period evolution, which make different assumptions, and one of those models is the one I just presented. And another one is a model where the authors assume a stable environment, and you also see this delayed sensitive periods because of an incongruency between the early ontogeny cues and the prior. So it was a specific manipulation that the authors in this model used. But what this indicates is that non-early sensitive periods may be a common feature of development, which might evolve across a range of environments, traits, and life histories. So I just want to pause and just put up what I think is at least one of the insights I would like you to take away from this talk, which is that changing environmental conditions, more so than stable environments, may favor sensitive periods beyond early ontogeny. And as I just explained, other models have also provided these kinds of insights. And recently I submitted a paper in which we synthesize findings from both mathematical models and empirical papers which demonstrate sensitive periods beyond early ontogeny. On the bottom of the slide, you can see a link to the preprint in case you're interested, but I'm also very briefly going to sketch out what we've observed in that paper. So in that paper, we observed that sensitive periods for the development of social behaviors in mid-ontogeny or adolescence, if you want, occur often in mammals, especially in humans and rodents. So we hypothesized that this may be due to increases in the value of social information. As organisms start to navigate novel social landscapes during adolescence, they gain independence from their caregivers, and they can sample cues that are more relevant to them. We also observe sensitive periods towards the end of ontogeny across various invertebrate species, such as snails, wasps, water fleas, and bulb mites. Here we hypothesize that changes in the physical and social environment of these species may favor phenotypic adjustments towards the end of ontogeny to prepare them for the subsequent developmental stage. So this kind of concludes the first part of my talk, and the rest will hopefully be a bit less dry. Earlier in the talk, I promised to come back to individual differences. More precisely, I wanted to talk about what these evolutionary models can possibly tell us about differences between individuals of the same species. Okay, maybe it does get more dry again. In this context, it is important to note that the type of findings I presented throughout my talk are typically at the level of species differences. So that is, the results indicate what patterns of plasticity may emerge for species that have evolved with specific priors and cue reliabilities. So here you can see the actual findings from my variant cue reliabilities model, and each quadrant is a population of species, if you want, corresponding to a unique combination of prior and cure liability pattern. But the models also lend themselves to dig deeper into individual differences. What you can see here is the optimal policy that underlies the derived patterns of plasticity for a specific combination of prior and cure liability. And remember that pattern of plasticity, I guess you can't remember because I haven't told you, but the pattern of plasticity is actually an average, across the population members. So these policies display the optimal developmental trajectories of the entire population for given prior and cure liability. And if we look at this, we can gain some insights about individual differences, both in phenotypic development and patterns of plasticity. Actually, Frankenes and Panchonathan, who started to develop these specific models of incremental development, noticed really early on that in these models, individual differences emerge from stochastic sampling. As I explained earlier, in the models, organisms learn about the environment through cues, and the cue reliability indicates how reliably cues predict the true state of the world. So due to chance, some organisms may sample more consistent sequences of cues and others more inconsistent sequences of cues. So what you can see here on the right, and the resolution is not great, I apologize for that, is a optimal policy for some model, for some parameter combination. But what I would like you to notice is, so whenever this brownish line goes up, organisms sample one type of cue. When it goes down, they sample the other type of cue. The colors indicate what kind of phenotypic decision organisms make. So what you notice is when it consistently goes up, 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 or down, 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 or mainly up, or mainly down, it's always the same color. So organisms consistently specialize towards one type of environment. However, if cues go up, down, or some other pattern, then we get like this variability in phenotypes that they develop. And this stochastic sampling can result in individual differences in phenotypes and in patterns of plasticity. It turns out that this finding is quite robust across models, making different assumptions, including those that I presented earlier in this talk, so I can observe that same pattern in my optimal policies. Leading to key insight number 2 that I would like to highlight, which is that stochastic sampling can result in individual differences, both in phenotypes but also patterns of plasticity. Earlier in the talk, I explained that models have shown that plasticity declines more slowly when experiences are noisy and do not help to reduce uncertainty about the state of the environment. This finding is both true at the between species level, but also at the between individual level. So if you have populations that in the model, I keep touching this, that in a model have evolved with a lower cue reliability, then just by virtue of that lower cue reliability, cues will tend to be more noisy because the information is just less good. So for species like that, in the models, we observe that plasticity is prolonged. Within a species, we also see that in the optimal policy, that when individuals receive more inconsistent cues, that plasticity is also prolonged. So this is again the effect of stochastic sampling. And empirical studies actually support such an explanation. Studies in humans, birds, and rodents have, for example, shown that noisy, inconsistent inputs tend to prolong sensitive periods in individuals. The idea of stochastic sampling also provides potential explanations for the puzzling observation of individual differences in otherwise genetically identical organisms. A mechanism which is often implicated in the development of individual differences are feedback loops between an individual's behavior and their state. The state can include their physical features, their personality, their social rank, but also what they know about the environment. When individuals express different behaviors during development, this can cause changes in their state, which in turn can shape subsequent behaviors and so on, so creating this loop. Positive feedback loops can then increase individual differences, so creating more variability, while negative feedback loops might lead to behaviors which make individual states converge again. Stochastic sampling of experiences is an additional component that can interact with this loop between behavior and state, further amplifying individual differences as a consequence of sampled experiences. If you're interested to learn more about the kind of theoretical evolutionary underpinnings of these feedback loops, I can recommend a recent model that is building on previous models of incremental development and provides some insights into the selection pressures which may result in positive and negative feedback loops. So I'm just putting this up there in case you're interested. The last thing I would like to address is the unfolding of individual differences across time. So in the models, I mainly focused on differences between species. However, in my variant Q reliabilities model, I dug a little deeper into the development of individual differences. Specifically, I looked at the development of rank order stability to understand when during ontogeny individual differences develop and whether they maintain stable or can still change. For this purpose, I simulated populations of individuals and I started to rank them according how specialized they were towards one environment. So the most specialized individuals at the top, and then it goes down the list. In this plot, I only want you to focus on the black bars. So they indicate the proportion of rank switches that occur within the population of simulated organisms between two consecutive time periods. So when the bar is high or large, what this indicates is that from one time point to the next, organisms switched in rank quite a bit. So that means that individual differences have not stabilized yet. So the population is still kind of, figuring out their rank if you want. And the smaller the bars get, the more stable these individual differences have. So like the top individual will remain at the top, the bottom individual will remain at the bottom. We can compare these bars across the different quadrants, again, to get species-typical insights, but we can also focus on individual plots in order to get insights about what's going on in that species. In the leftmost column, we observe a common pattern in the empirical literature suggesting that individual differences develop and stabilize over time, causing age-related increases in so-called trait repeatability, which is the extent to which individual differences in trait scores are maintained over time. However, in my model, individual differences don't always continuously stabilize across ontogeny. We observe that in some environments, ranks within a population might first destabilize before stabilizing. My model suggests hypotheses about the selection pressures that can result in the more common pattern of increasing trait repeatability and in the less common pattern of, decreasing and then increasing trait repeatability. There also exist empirical studies suggesting that individual differences might not always stabilize continuously across ontogeny. For example, in a paper from 2015, Würz and Krüger observed large variation in repeatability across different traits and life stages in zebra finches. Some traits showed no repeatability, while others were only repeatable in some portions of ontogeny. So the authors suggest that trait repeatability may often be overestimated in the literature because studies tend to only measure individuals at two time periods, which is very understandable. It's extremely expensive to conduct studies like that. But the idea is that when we include more time periods across ontogeny, we might not always find the assumed consistency of individual differences. And recent advancements in animal tracking, I think, outline a very promising future for getting a little bit to the bottom of this because they make it easier to continuously track behavior in individuals and then to see how traits unfold across time. This kind of also brings me to my conclusion. Clearly, my work involves a very theoretical approach to understanding development and changing environmental conditions and the development of individual differences. But I do think it's extremely important to kind of ground these results and try to test them empirically in some way or shape or form. Thus, future work may empirically test these different reasons for sensitive periods at later life stages and the development of individual differences in various taxa, such as invertebrates, insects, rodents, and fish. And I think that understanding why and when sensitive periods and individual differences in plasticity evolve can advance our understanding and contribute to an integrative science of sensitive periods, which maybe down the line could advance human research. Last but not least, I wanted to thank my advisors and collaborators on these projects, Willem Frankenhaus and Karthik Panchanathan. And obviously, I want to thank you for listening to this very dry talk. I'm excited for your questions, and feel free to email me if anything comes up. Thank you.